Welcome to another episode of Inside Chlorpromazine. I'm your host Sowmya, and today we are exploring something fundamental to how viruses infect us, the point of entry. A topic, the gatekeeper, how chlorpromazine could block viral entry. We'll examine how SARS-CoV-2 gains access to our cells and a compelling hypothesis that chlorpromazine, originally an antipsychotic drug, might act like a bouncer at the door, proving that entry. To help us understand this, we are joined by Chythanya, a microbiology researcher who specializes in host-pathogen interactions. Welcome, Chaitanya.
Thanks for having me. Excited to talk about this topic. It's one of the more fascinating mechanisms we investigated.
Let's start with the basics. How exactly does SARS-CoV-2 enter human cells?
Sure, the virus uses its spike protein to bind to a receptor on the surface of human cells called ACE2, angiotensin converting enzyme 2. The receptor is abundant in cells lining the lungs, intestine, blood vessels, and even in the brain. Once it binds to ACE2, the virus can enter the cells in two main ways. Direct membrane fusion, primarily in lung cells with TMPRSS2 protease. Endocytosis, the cells engulf the virus into a vesicle-like swallowing it. Endocytosis is especially important in tissues where membrane fusion isn't dominant. That's where chlorpromazine comes in.
Could you break down endocytosis for us in simple terms?
Of course, endocytosis is like the cell creating a pocket in its surface to wrap around something. In this case, a virus. Once the virus is enclosed, it's brought inside the cell in a little bubble called an endosome. There are different types of endocytosis. One subtype called clathrin-mediated endocytosis is especially important for that entry of many viruses, including SARS-CoV-2 under certain conditions. This process uses a protein called clathrin to form a coat around the vesicle. Think of it like packaging. And here is the key. Chlorpromazine is non-inhibitor of clathrin-mediated endocytosis.
So, how exactly does chlorpromazine block this process?
Chlorpromazine disrupts the formation of clathrin-coated peaks. It causes clathrin and its adapter proteins to relocate from the membrane into the cytoplasm, where they can't do their job. When this happens, the virus can still bind to the cell surface but can't get inside, at least not through the clathrin dependent route. For viruses that rely heavily on the pathway, it's like shutting the door in their face.
What's the advantage of blocking the virus right at the point of entry?
It's one of the most effective strategies in antiviral therapy. If a virus can't enter the host cell, it can replicate, mutate, or cause disease. You're stopping the infection before it even begins. Also, targeting host mechanisms like clathrin-mediated endocytosis reduces the chance that the virus will quickly develop resistance. Most antiviral drugs target viral enzymes which mutate quickly, but cellular pathways mutate much more slowly.
So, why aren't we already using chlorpromazine widely as an antiviral?
It's a good question. While the lab data is promising, clinical trials are needed to verify whether the drug's antiviral activity translates into real patient benefits. The challenge is finding a dose that's both effective and safe, especially since chlorpromazine was designed for psychiatric use. Another factor is tissue specificity. Some cells use clathrin for virus entry, while others may prefer membrane fusion. So, Chlorpromazine might be more effective in certain tissues like the gut of brain than in lung cells. That said, it's been seriously considered in several repurposing studies.
Could chlorpromazine be used in combination with other antiviral agents?
Absolutely, that's very attractive idea. You could use chlorpromazine to block entry and another drug like remdesivir to stop replication. It's a multi-step blockade, like locking both the door and the safe. This combo approach could be particularly useful in high-risk patients or in long COVID cases where viral persistence is suspected.
To wrap up, chlorpromazine might not be the ultimate antiviral, but its ability to block viral entry, particularly through clathrin-mediated endocytosis, makes it a compelling candidate for further research.
Exactly. It's a great example of using older, well characterized drugs to meet new challenges. And entry inhibition could be a game changer, especially in combination therapy.
Thank you, Chaitanya, for guiding us through this fascinating aspect of viral infection and pharmacology.
Always a pleasure.
That's it for today's episode of Inside Chlorpromazine. Join us next time as we look into chlorpromazine's role in modulating cytokine storms and whether it can help calm the immune system during hyperinflammatory phases of COVID-19. Thank you.